from the black hoodie stain swabs, it was approximately 460 times more likely if it originated from jarvisbutts and an unknown contributor than if it was from two unrelated unknown contributors. And this provides moderate support that jarvisbutts is a contributor to the black hoodie stain swabs.
Okay. And you can't say what that stain was, can you?
No.
Okay. And then you said you did one for jarvisbutts, and you did one for naziyahharris.
Correct. So when comparing naziyahharris, it's approximately 3.7 million times more likely if it originated from naziyahharris and one unrelated contributor than if it were from two unrelated unknown contributors. And this provides very strong support that naziyahharris is a contributor to the black hoodie stain swabs.
And in interpretation 3, this is the second of the black hoodie stain swabs, 5BD. When they're separated like that, when you have like 5BD and 5AD, does that tell you that they're different parts that are being swabbed, different areas?
Generally, it will be two different stains on the same item, but the person that actually took those swabs, they would best be able to describe where they were collected from.
Okay. So as it relates to 5BD, the black hoodie stain swabs, what did you find there?
That was one individual in that sample, and it genetically typed as So I determined that this was from 3 individuals. There were 3 individuals in that sample, and at least one of them genetically typed as male. And when comparing it to jarvisbutts, it was approximately 2.1 million times more likely if it originated from jarvisbutts and 2 unrelated unknown contributors than if it originated from 3 unrelated unknown contributors. And this provides very strong support that jarvisbutts is a contributor to the black hoodie swabs.
And what about the comparison to naziyah?
When comparing naziyahharris, it is 97 sextillion times more likely if it originated from naziyahharris and 2 unrelated unknown contributors than if it originated from 3 unrelated contributors. And this also is very strong support that naziyahharris is a contributor to the black hoodie swabs.
And how many zeros after your sextillion?
Sextillion is 21. And just for reference, anything over 1 million is considered very strong support.
Okay.
Pink bodysuit stain swabs, pink bodysuit swabs.
And your next interpretation is to the pink bodysuit stain swabs. And you perform this assuming that there was
Correct.
Tell me your comparison with Jarvis butts.
So it was 24 trillion times more likely if it originated from two unrelated unknown contributors than if it originated from Jarvis butts.
Is where the positive phenol stain was located. This would be the interior back portion of the item. So ones that had a fluorescence to them with a silver Sharpie, so I could further test after done screening. And IR, which is an infrared, is similar to, it's also an alternate light source, but it's more so for blood on dark items where the dark fabric would become lighter, and if there's blood or possible blood present, it would appear darker. Anything that I saw, I marked with a yellow paint pen with IR next to it, so I can distinguish between the ALS samples, the ALS areas that I saw and the IR areas that I saw.
Okay, and we see a lot of markings on the front of this hoodie.
Yes.
Are you able to tell us, and I know you said the color, but what portion of this is the IR versus the ALS? And it may be easier for you to step down, although there's not really room.
So the ones that I observed with IR, I actually wrote IR next to them. I don't know if that's visible on there, given how small the picture is, but other photos, I took pictures of
Close
certain
enough.
areas, yeah.
And if we could just go to the next, or if you
Correct, where there's, like where the icon, the hand, oh, it moved. Right there, and there's an arrow pointing to that large area, like those were areas that dropped out under IR, so I put IR next to it so I'll know what to follow up and how to follow up.
And that IR is to indicate blood, correct? It's not a test for blood, but
It's a screening tool used.
and this is a closer view. Is this after you've done any other testing other than the light screenings?
Yes.
Okay, what testing was done at this point based on this photo?
This is after the pheno testing was done. This particular picture is reflecting a phenopositive area. And it was also screened with IR. As you can see right over the swabs there, it says IR. And that's what led me to follow up with pheno, was because I saw it under IR and it was phenopositive.
And where is this, if you're able to say, on that hoodie?
So this would be, if I was wearing it, it would be in this area, the upper shoulder,
And for the
bicep
record-
area, yes.
For the record, the witness indicated to her right upper shoulder. Yep, right. And what are we looking at here for this photo?
Those are the cartridges used in the hemotrace test. As you can see, you read them per line. The first two only have the one line, that's the control line, which, because there's no visible line in the testing area, it is a negative result. And the third through fifth cartridge have the double line, so those are considered positive results.
And that's for stains, the stain numbers are listed there, correct?
Correct.
And that's stain 20, 28, and 38 that were positive on the hemotrace test?
Yes.
Going to slide 23. And what are we looking at here in this photo? And at what point in your testing was this photo taken?
This was after pheno testing and the AP testing, which was the semen testing. And I put the key there because these are for the results that I wrote on the sweatshirt. So my tech reviewer and whoever's reviewing the photo would know what those meant when they saw them on the sweatshirt.
Over time and in certain conditions, sure.
Weather, stuff like that,
Potentially as sunlight can degrade DNA.
the elements in general, bacteria.
Yes.
Okay. And so when it degrades, does that mean that the DNA molecule itself is breaking down?
Yes, it's degrading, yeah.
And what is the impact of that on a DNA test?
It could just be lower than it would have been. So there's still a profile, it's just lower. We also see a very specific pattern when something is degraded, where it, instead of peaks kind of being nice and straight, they kind of ski slope off. So we can identify it.
Okay. Could it mean that you get an incomplete or a partial profile?
Sure, yeah.
Was that a factor at all in any of the swabs that you tested in this case?
I didn't see any very specific that pattern that I was talking about. I didn't see that. Some of the samples are lower, so they may have been possibly, if they were in the elements, degraded or diluted.
Okay.
We went out once. It was probably maybe a couple weeks later that we went out there. We went out there once, and I remember it snowed. So when we went out there, it wasn't as easy to search. We're kind of looking if somebody was just placed out there, or if something was easily identifiable, but because of the snow, we knew that we were gonna have to come back again once that melted, because it'd be easier to see underneath.
Okay. And so do you go out there again?
Yeah, we've been out there multiple times. I've probably been out there 7, 8 times, all with various dive teams. We searched the complete river from 8 Mile all the way down past 96 on foot with cadaver dogs, in boats, in the water. There's several log jams. We had state police actually had their helicopter fly the entire route so we could GPS all the log jams. We had to coordinate with specialized teams to come in to help cut these log jams apart, which go throughout the river. And then throughout the season, obviously we had some rain, so the water goes up and down, which now brings the debris onto the land. So you have to actually search the land a couple hundred feet, and in some places that the water has pulled debris.
So after you recover the pink jumpsuit, the black hoodie, the shoes, what do you do with them?
Those are all, well, the evidence techs collect them. They're placed on evidence. Those items were wet, so I had to wait, obviously, for them to dry. Once they're ready to be processed and they're finished drying, then I create a lab report. And items are selected and turned over to the state police lab for processing to look for any type of DNA evidence.
Okay, so once those are placed on evidence by the lab tech, you're the one who then submits them to Michigan State Police.
Yes.